Hey friends, welcome to the Lyme Insider podcast. Today we have an amazing episode. I'm joined by two amazing researchers, and our primary guest is Dr. Choukri Ben Mamoun. He's a PhD researcher, professor at Yale University, and he's the mastermind behind one of the most recently exciting papers that I've read called Tafenoquine-Atovaquone combination achieves radical cure and confers sterile immunity in experimental models of human babesiosis. So this is an amazing topic, a great paper. We're going to have the paper in our show notes, but this is a great conversation. And we're also joined by special guest, Dr. Tim Sellati, PhD, who's a borreliosis researcher, who's now the Chief Scientific Officer of the Global Lyme Alliance. And together, we're going to be unpacking what this paper really means from a scientific perspective, and also how we may be able to interpret the findings and put this into clinical practice. So stay tuned and enjoy this episode. Well, hey guys, thanks so much for joining us for the podcast here and getting together so that we can really shed some really great light on the great new work that's been done. Choukri, your lab, and Tim, I think it's so important for the three of us to get together to bring our varied viewpoints so that we can share with everybody how we can actually move forward and use this what I find is tremendously valuable information, but in a responsible and truly scientific way so that it can lead what we do clinically and scientifically. So thanks so much for joining me.
Thank you for having us.
Thank you for having me.
Yeah. So Choukri, I think maybe we could just start off with could you give us maybe a brief overview of the study and maybe some of the key findings so that we can use that as our jumping off point?
Yeah, absolutely. Research which was published in the Journal of Infectious Disease this year, 2024, and also made the cover of the journal just about 2 weeks ago, focuses on the use of a combination therapy consisting of atovaquone and tafenoquine as a way to eliminate Babesia infection. This was done initially in vitro using Babesia duncani as a model system because it's really an excellent system to study intra-arthrotic parasitism. Then subsequently, once we have demonstrated that the additive nature of the combination between the two drugs, we went into animal models of babesiosis, in this case, Babesia microti, using the immunocompromised mouse model, in which the microti causes a rapid increase in parasitemia or number of red blood cells that are infected with Babesia microti, and that plateaus, so that Babesia microti does not cause death in animals, but it causes this high parasite burden or parasitemia burden. The second model is Babesia duncani model, and that is a lethal mouse model or mouse model of lethal infection mice, especially C3H mice with Babesia duncani at low dose below 10 to the fifth. The mice will develop very low parasitemia, but they will succumb to infection due to a major cytokine storm. Whereas if you infect them with a very high dose, this is in the 10 to the 6, 10 to the 7, they die from a very high parasitemia in a very short period of time. So in both cases, they serve really as an excellent model to evaluate the efficacy of any therapy that we study. And in particular, in the case of duncani, if your therapy is effective, you're going to see animal survival. Whereas with the microti, you're going to see clearance of infection or reduced parasitemia, but it never tells you really whether you might have relapse later on because there is no deleterious effect on the animal itself. So that's the two models that we used with atovaquone and tafenoquine to demonstrate that the combination itself was superior to any single drug treatment. And the combination resulted in elimination of parasitemia in both animal models and helped animals survive the infection, and that was for a long period of time. A second major finding in the study, which was really very unique to this work, and that is because C3H mice are immunocompetent mice, we were able to demonstrate for the first time that this clearance of infection also resulted in protection of the animals from subsequent challenge with a lethal dose of infection. To make that clear, because the drugs were so effective in clearing and killing the parasite, we believe that the material that was released from the parasite served as antigens, as sort of a vaccine that mounted immunity in the immunocompetent mice. That immunity was lasting for a significant amount of time, that when we took those mice, after treatment has ended for several weeks, And we took those mice and challenged them with a high dose of Dankani that will normally lead to that within 6 to 8 days. In this case, these mice were completely protected. There's no treatment here, just the fact that they have developed immunity. It's basically now become not only treated mice, but became vaccinated mice. And they were completely able to clear the infection. That was a major finding that shows that this combination therapy has really what you would refer to as the 1-2 punch strategy, which is really clearing the infection and also providing protective immunity from subsequent infection. That's in a nutshell what we have reported in that paper.
That is so incredible, because I'm like, there's so many places where I want the train to stop along the tracks and different stations so we can talk about it and unpack it. I mean, there's so many things. You know, Tim, when one of the roles that you're playing at GLA is not only like as the chief scientific officer, kind of from, you know, communicating with researchers and such like Choukri, but and getting the funding together and evaluating all that, working with clinicians like myself to, you know, figure out what we need from a clinical sense. But really, I think it's the education of the clinician and the patients and the public to understand what this research means. And when you read the reports, you know, and hear the summary that Choukri is just sharing, what kind of pops out for you as like the major pieces? And maybe where have you seen some of the public or doctors kind of grab some of these really cool things like sterile immunity and this thing and that thing or complete eradication, and kind of maybe start heading down a path that maybe the science doesn't quite lead us down just yet?
Yes, it's a fantastic question. I think what most intrigued me about this study is what Choukri was just discussing, the idea that drug treatment of the Babesia infected mice could confer protection against subsequent reinfection. And when we think about a primary infection, the first time someone is infected with a microbe, this often results in the generation of long-lived memory B cells that are responsible for producing antibody, and also memory T cells that actually enhance that protective immunity against subsequent infection. So what I was amazed by is the fact that the combination drug-induced killing of Babesia actually confers protection against subsequent infection. And I was curious to know, Choukri, whether you think that's because the combination drug therapy actually stimulates better protective immunity in terms of memory B and T cells than a primary infection does.
Yeah, so this is a very important question, and one that we are still going to be investigating very carefully. So we know in the case of Dankani, that's very difficult to investigate because once they are infected, they succumb to infection. That's a very severe model of infection. But if we were to use a different model, and we have done some of these experiments, it will get too complicated to explain here, but we can use a different mouse model that survives Dankani infection, and we can infect them with the parasite and then challenge them with a very high dose of infection. And in that model, without treatment, we also see clearance of infection. So in both cases, what we think is happening, whether for a mouse that's capable of killing the parasite or in a mouse that's not capable of clearing the infection, but the treatment aids that animal in clearing the infection, both achieve the same goal, which is to release the parasite antigens that then trigger a sort of immunization similar to what you would do if you were to vaccinate individuals, say for COVID-19 or for measles or other stuff. Right. So you're releasing basically an attenuated parasite, or just the antigens released from the parasite. As you probably well aware from what I presented last year during the keynote lecture, we've discovered that the Babesia parasites have this unique vesicles that they produce and excrete into the host. And those vesicles are full of parasite antigens. It is the most likely scenario, and we think the hypothesis we have here is that when we kill the parasite, either with treatment or as a result of a strong immunity for some animals, you get this release of those vesicles that are fully charged with several antigens, and that is what's triggering the immunization in animals. Mm-hmm. And in the course of that study, and you can see it in one of the last figures in the paper, where we monitored the antibody production over time. And it was remarkable, I think, consistent with what you just said, Tim, about memory B cells and T cells. Right. We saw, and you will look at it, it's really beautiful data that I was really surprised when I saw it initially, that before the challenge, we could see increase in the antibody titers in the animals over time. And that was the reason why we decided to challenge those animals. But then we challenged them with a lethal dose, with a very high dose of infection, and they cleared the infection entirely. So we decided to look at the antibodies in these mice after challenge again. And the titer went even higher, which tells you that those memory cells were ready now.
Yes.
That's what you would do with any vaccination without really doing an antigen-based vaccination or an attenuated parasite vaccination type. You really have a treatment that did the work for you from the get-go.
No, it's fantastic. And so, Tom, I want to come back to the tail end of your question. Choukri just answered it beautifully, is the idea that, yes, we've got this incredible study now that talks about the power of dual combination drug treatment, but there are still a lot of questions to be answered about exactly how this works. Choukri loves mouse studies. I love mouse studies. I did a lot when I was an active researcher. There's an incredible amount that we can learn about human babesiosis, or in my case, Lyme disease, using mouse studies. But at the end of the day, a mouse is a mouse and a human is a human. So we can't too quickly jump from what we've learned from how to treat mice to how to treat humans, because we don't know if it could harm humans, where it's obviously not harming mice, it's protecting them, or we could treat humans in a similar fashion, and it may help some patients, but not all patients, maybe very few patients. And we just don't understand why that's the case. So we have to have a solid foundation of knowledge that comes from basic research and translational research using animal models of infection before we jump into physicians using these in patients.
Yeah, I mean, I think your points are both really well taken. And one of the things that I see is that I think it's great to ask you what you think is the most important part of the paper, because I can guarantee you almost every doctor and patient listening to this did not think of what you two just said. We weren't thinking about the sterile immunity. We weren't thinking about this sort of like essentially autologous vaccination, vaccination through exposure type of thing. We're just like, hey, how do we kill it and make people better? And it's really interesting to me, like where we focus, which is why I wanted to have this conversation, because I think that we've already used a few words and talked about a couple of things that people get really, when we're trying to take the research and put it onto the, let the rubber meet the road, so to speak, in a clinical practice, we hear a few words in here. And so I have a few notes, so we'll walk through them. But Choukri, one of the things I know that the three of us have talked about before when we were kind of like planning to do this is like the immunocompromised model. So like, I think a lot of people with Lyme disease say that they're immunocompromised. And the truth is the vast majority of them may be immunochallenged. They may be a little functionally immunosuppressed, if you will. But from a medical scientific research perspective, they're not truly immunocompromised. So could you tell us maybe briefly what an immunocompromised mouse is, but then also why do you start there, not just with sort of a quote unquote normal mouse? Because I think that that's an important piece for physicians, mid-level healthcare practitioners, and the public to really understand.
Yeah, that's a great question. And I can only speak for Babesia infection, not Lyme, because the infection is quite different. I was really hoping initially to tell you a little bit about the parasites, because that's a very important
Feel free, yeah.
in this entire concept. Babesia parasites are what we refer to as apicomplexan parasites. They are protozoan parasites. They are eukaryotic pathogens, unlike Borrelia, which is a bacteria. So this is very important to keep in mind. So Babesia parasites are apicomplexan protozoan parasites. They belong to the apicomplexan phylum, which is basically the same phylum that encompasses other parasites that infect humans, including Plasmodium falciparum that causes the most severe form of human malaria, or Plasmodium vivax. It also includes the Toxoplasma, Cryptosporidium, and other parasites. That's very important. And they all share in common this important feature of the parasite itself, which is this apical end that allows the parasite to invade the host cell. That's a really an important criterion to think about. And that's in the case, for example, of malaria parasite, that apical end is critical for the parasite's ability to invade hepatocytes, liver cells, then to invade the red blood cells, then even when it gets into the mosquito, to traverse different tissues to get into the salivary glands. In the case of Babesia parasite, that apical end is also critical for the invasion of red blood cells, and most likely serving also a role in the tick transmission of the parasite. And so when you think about it this way, you have to think about these parasites actually residing specifically in red blood cells. Red blood cells don't express MHC class 2, and so it's an ideal system for parasites to hide from the immune system, if you wish. So there are other ways for the immune system to clear those infected red blood cells, and one of them is really the spleen clearance. And that's why in whether animals or humans, you see this enlarged spleen as a result of the infection with Babesia or Plasmodium. It's because there is this huge amount of red blood cells that are being cleared, often because they cause a change in the morphology of the red cell, of the infected red blood cell. Interestingly enough, not only the infected red blood cells that get cleared, but also normal red blood cells. And that's why you see sometimes patients with very low parasite load or low parasitemia, and yet they're anemic, if that makes sense, because you're clearing a huge amount of red blood cells. So to come back to your question, spleen plays a major role in the control of the infection. And so if you have an individual who's splenectomized, that individual now is not capable of clearing the infection, and that's really one of the major risks of Babesia, along with cancer patients who can be immunocompromised as a result of treatment, let's say with rituximab and other drugs of that sort. And then, so here we are talking about serious alteration of the immune system in these patients, and as a result, it makes them highly vulnerable to infection with Babesia parasites. So that was to make clear. In the case of the mouse models, in the case of microti, which is the predominant form of Babesia in the US and also other parts of the world, the immunocompromised mice that we use are either SCID mice, they carry a mutation that alters their immune cells. We also use RAG knockouts, which are also genes involved in the pathways that regulate the immune response to pathogens. In both cases, once you disrupt that process, we see finally that Babesia microti is not cleared anymore. You see this high burden. In fact, in the case of immunocompromised mice following infection with microti, we can see as high as 50, 80% of parasitemia, which means that 50 to 80% of red blood cells are infected, and yet these mice don't die.
Yeah.
Right? And that is really critical. But if you were to take Babesia microti and infect immunocompetent mice, bulb C mice or C2H mice, etc., you get this initial peak of parasitemia depending on the dose. But then it's cleared immediately by the immune system.
Yeah.
Right? So that's why we utilize the immunocompromised mice, not only to serve as a model for what we see in patients who are immunocompromised and usually get the high burden of parasitemia. And in some cases, if that parasitemia level gets above 8%, 10%, 11%, they have to undergo exchange transfusion. That's why we use that animal SCID model and the RAC2 knockout model to mimic that profile.
Right.
If we do it in immunocompetent mice, you have to do a study very carefully, because you will, let's say you inject with a high dose of the parasite, you will get a very high peak of parasitemia, and then the immune system does it. So when do you measure the efficacy of your treatment?
Yeah.
See,
Right.
you have really to time it very well. And you can see that the peak is no longer up there, it's slow or inexistent, and you know your drugs work, but you will always be challenged by the fact of the question similar to what Tim asked earlier. Is it your drug that's doing it, or your drug stimulating the immune system to clear your infection?
Right.
Mhm.
That's the advantage of using the immunocompromised animal model to study these infections. In the case of Duncani, and you can also look up our paper in 2022, which is a really beautiful paper. The entire focus of that paper was just to optimize the animal model so that all labs around the world can use it. And in that case, we did all doses of the parasite in immunocompetent as well as immunocompromised mice. And as I told you before, Duncani causes lethal infection in immunocompetent mice. If you were to do the same infection in immunocompromised mice, now they die primarily from hyperuricemia and anemia. They don't die from the cytokine storm. So you can see the importance of the immune system in the control of Babesia infection just from these various models.
Right. So, Tom, I want to just jump in for a second and highlight 2 points that Choukri made, because I think it will be of interest to your audience. One is where mice and humans do sort of act the same way. We talked about normal mice that are immune competent. They're competent to protect themselves against Babesia infection. And that when you infect an immune competent mouse with Babesia, oftentimes it's able to control that infection, kill and clear the parasite. Interestingly, in the human population, as many as 30 or 40% of individuals that have antibodies against the Babesia parasite, they are asymptomatic. They essentially were infected with Babesia, but they don't have clinical evidence of disease. So that's where there's a similarity between the two. Another interesting difference between Babesia, which is a tick-borne infectious agent, and Lyme disease caused by Borrelia burgdorferi is when you're infected with Borrelia burgdorferi, neither the primary infection with the Lyme bacteria nor antibiotic treatment of an individual that has Lyme disease will confer protective immunity against reinfection. You can be infected with Borrelia over and over and over again. And treatment doesn't enhance that protection as it appears to in the Babesia infected mice. So that's a very striking difference. So not all tick-borne infections are the same. Along similar lines, if you were to look at the bacteria that causes Lyme disease and the bacteria that causes syphilis under a microscope, unless you know what to look for, they look remarkably similar. But obviously, from a clinical perspective, the similarities between Lyme disease and syphilis sort of end at the microscope. They are very, very different infectious diseases, and they have to be treated differently. So I think it's very important for physicians to understand that if they have a patient that they suspect has a tick-borne disease, don't just think about Lyme disease. Think about Babesia as well, and not just Lyme and Babesia, but anaplasmosis, ehrlichiosis, Powassan virus. So you really have to listen to the patient in terms of the symptoms that they're presenting with, base understanding of the different tick-borne diseases and how they might present clinically to help you with that differential diagnosis.
Right. Yeah. It's so interesting too, because I think that I almost in the back of my head, just being kind of, feeling a little goofy today. I'm like, wow, I could just go get infected with Babesia Duncani or one time and then get it treated and I'll be good to go for the rest of my life. Probably not what we want to be doing. But one of the things that comes up so much in the work that we're doing, guys, is we try to take the research and all this amazing knowledge that you're, and Choukri, like the work you guys are doing in Babesia is so required, because I remember years ago, I was working with iLads to create the fundamentals, in tick-borne illness so that people could better understand symptoms, better understand sort of the pathophysiology of the organisms, and how they affect patients. And then how do we go about treating people? And one of the things I came across, even in the, around 2010, 2014, everybody was still saying we know so little about the Babesia parasite in any model. And so the fact that you guys have been at the forefront of kind of changing that, or at least, I mean, we were talking about so little known in the veterinary model. And then now I feel better because I'm learning more about how this parasite actually works in humans, so that I can do what Tim was saying, is like not only look at the person as a whole, but then piece out what might be Babesia, what might be, Lyme disease, or what might be other, which I think is always, as a physician, I was always told, keep your blinders off. Don't just go, oh, I have like one Babesia infection I found, and let's pretend the rest of life doesn't happen. So when I'm trying to take the information in your paper, one of the big questions, and put it into clinical practice, one of the big questions I have is, what about tafenoquine by itself, and why were we combining tafenoquine and atovaquone together? Because I know that there was definitely some great findings with tafenoquine by itself, but you guys are really focused on the combination. So maybe you could walk us through that, what tafenoquine did by itself and why the combination really was looked at, and how it turned out to be different than the results as a single agent.
Great comment, Tom, and I wish I could comment on the first part of your discussion about Babesia, because I have a lot to say on that topic as well. But let me talk about the focus of this podcast, which is about the combination therapy. We didn't include the combination therapy until we tested the single drugs, and the single drugs have already been part of the clinical treatment of babesiosis. So atovaquone has always been part of the combination with azithromycin in the treatment of human babesiosis for years. And we know that there is relapse caused as a result of this treatment. So treatment failure is very common with atovaquone azithromycin. In fact, in our animal models, and this was published in our first paper in Journal of Experimental Medicine in 2016, where it was the first time we took these drugs and we tested them in our animal model of infection. And I can tell you, atovaquone, without failure, we always see a relapse in vitro, in animals, always. It's almost as if the mutation is there before we even start the experiment. So every single experiment where we treat with atovaquone alone, you will see a relapse. What is surprising, and this is another topic, and I hope it doesn't distract us from the main topic, it has, when we get this with this what you would call resistant parasite, but just really relapsing parasites, when we sequence them to see the mutation in the cytochrome B gene, which is the primary target of the cytochrome B, or at least the enzyme encoded by the cytochrome B gene is the primary target of atovaquone. In some cases we see the mutation, in some cases we don't see the mutation. And that those parasites that don't have the mutation are actually sensitive to atovaquone. So this raises the issue as to whether atovaquone in some cases is just causing cytostatic effect in some of the parasites, or that we have some form of dormancy in Babesia that's actually not very well characterized and studied. And we have some evidence to suggest that there is some that some form of dormancy might exist in these parasites as well. So that's the first question about atovaquone. Tafenoquine was introduced to the clinic and has been used for treatment of babesiosis by some clinicians. And we were approached about this when we were in the middle of our study by physicians from New York, telling us that they had patients that failed the treatment. And there was a report last year, I believe, that showed that a patient after treatment with tafenoquine failed treatment. It seems very clear that it's the way to go. And in our studies in mice, we can replicate the same thing that we hear about in the clinic, which is that monotherapy with atovaquone or tafenoquine can result in relapse. We saw it it's presented also in our paper with 2 out of 6 mice treated with tafenoquine alone relapse. So when you treat with combination therapy, in this case, it's based because both drugs are additive, you're getting radical cure. And that's because you're targeting more than one target within the parasite. And then by doing so, you're increasing the potency of both combinations within the parasite. That's why we recommend the combination therapy for any future clinical trials. I know that there are efforts to do clinical trial with tafenoquine alone. I would say it's interesting because clinicians have already been using tafenoquine, but it will probably be just waste of time, in my opinion, to do so. And that the major focus should be on testing the combination of proven combination and a very well-defined animal model to advance combination of atovaquone, tafenoquine for now, because those are the drugs that we currently have. Could we use other combinations of tafenoquine, say with azithromycin, clindamycin, quinine, proguanil, for example? In our hands, azithromycin, quinine, and clindamycin are not as effective, at least in the animal model. Now, the doses used in humans are very, very high. We cannot even achieve those doses in mice. So it's quite possible that at those high doses, they are doing their job, will challenge the specificity of that treatment at those doses that humans take. But atovaquone, we know, works despite the relapse. So if I were to choose amongst all the drugs that are currently in the anti-babesial armamentarium, I would say tafenoquine and atovaquone are the best drugs to move forward with, are the best combination to create to do any future clinical trial.
I'll just highlight two things and let you riff, Tim. One of the things is just kind of like, one of the things I think we want to come back to in a minute is dosing. I wanted to really highlight one thing you just said. Human standard, if I heard you correctly, and if not, please correct me, but standard human dosing is dramatically higher than anything that you're achieving in a mouse model, yet you're showing complete eradication with this combination therapy without recrudescence. So without relapse, yet as a clinician, I'm just highlighting for everyone listening, we are always taking the standard work that's being done in research lab and suggesting that we need to do double, triple, quadruple, tenfold, let's higher is better. And like, I just really wanted to highlight for people, like, the research is already being done at a lower sort of, if we were to do a ratio between human and mouse, relatively speaking, the mouse is getting a much lower dose than we're standardly giving in clinical practice, if I heard you correctly, Choukri.
Do you want to go, Tim, first, or do you want me to respond to that?
No, you can respond to that, and then I'll come back to the combination drug therapy idea.
Okay, so Tom, let me just answer that in the kindest way possible. Let me just say that most of these drugs that you're talking about that are used in the treatment of human babesiosis were not designed for treatment of human babesiosis. They were actually anti-malarial drugs that were used for treatment of human babesiosis, because if it looks like malaria, treat it with malaria drug. That's basically the concept here. And that's what we have done. And so as a result of this, we are just using doses that have been used in the clinic and approved by FDA for treatment of malaria, there is nothing wrong about that in a sense, because those doses have been evaluated for safety and efficacy in the case of Plasmodium. But in the case of Babesia, there was a clinical trial conducted by Peter Krause and others, published in New England Journal of Medicine many years ago. But that was simply to say, okay, this is the combinations that have been reported in malaria, let's test them in Babesia, and they work. No one has done a very detailed study to know what is the most effective dose for humans. It could be much lower than that. It might be higher. I don't know for those drugs. But I can tell you, as a basic scientist who worked with the parasites in vitro, and I work with the animal models, which are very well-defined models in the lab that follow the law and the rules of scientific method and statistical power. If I were to be given those drugs as numbers and choose which ones are efficacious and which ones are not, I would never use quinine, clindamycin, or azithromycin at all. Yet that's what we have in the treatment. Now, do they work in humans at that high dose? Again, coming back to the same point, it seems they work. Every clinician tells me that all the time. I'm not a clinician, but I follow the science that I see in in vitro and in mice. And as Tim said, mouse is not human, so I can't really say whether they are right or wrong. But based on the data we have, if I were to say choosing amongst the drugs that are effective in vitro and in mouse models, if I were to talk to FDA about what's the best combination to advance forward or the best therapy, today, what we have in our hands as approved, clinically approved drugs that can be repurposed, certainly Tafenoquine-Atovaquone will be much superior to anything that's being used currently. That's what science tells us.
So, Tom, the point I wanted to follow up on with what Choukri was saying about combination drug therapies and maybe other combinations would work as well is I've heard a lot of discussion of artemisinin being used to treat patients. And I was intrigued, Choukri, by some of the beautiful studies, the experiments you did and described in this paper. I learned, I didn't realize that tafenoquine is thought to have its effect by increasing the oxidative stress within the red cell that is infected by Babesia. So in essence, the drug may be causing the red cell to become a very inhospitable environment for the Babesia to survive in. And your studies, I think, very clearly demonstrate that that could be the mode of action. But then I also noticed that when you treated Babesia infected cells with artemisinin, you saw roughly 75% of those cells having increased oxidative stressful environment, whereas the treatment with tafenoquine had maybe 50% of the cells with that more inhospitable environment for the Babesia to try and survive in. Do you think artemisinin and atovaquone might be an equally effective or perhaps even more effective approach to treatment?
Yeah, so I just want to correct one thing here. Is the oxidative damage is caused within the parasite, but whether by artemisinin or by tafenoquine. And in fact, in our paper, we are also finding that atovaquone causes. That was really surprising to us, which could also explain why the combination of atovaquone and tafenoquine causes much higher oxidative damage within the parasite that kills the parasite. Now, coming back to your question about artemisinin in particular, we have tested a bunch of drugs from the same family, artesunate and others. They are not effective in our hands in vitro. Now, again, it doesn't necessarily mean that they cannot work in vivo, because there is another concept here to highlight, Tom, and that is in vitro, we can test, we can determine what we refer to as IC50 of a drug. You probably know this very well in bacteria and animals, and fungi talk about MIC50. So the IC50 value really tells us specifically how the drug is effective against the parasite, and it can tell us also many things about that drug. Not only the ability to target its specific target within the parasite, but also potential accumulation of the drug within the parasite. When you're treating animals, the dose that you're using is far higher than what you will ever test in vitro. So as a result of this, you will always have much higher concentration of the drug within the parasite than what you will achieve in vitro. And so it's quite possible that in vivo, artemisinin at a higher dose as you use in animals, if you were to combine it with atovaquone or tafenoquine, you might also have higher efficacy. But based on what we do in vitro, we try to work with in vitro data to guide our animal studies. If I have a compound that works effectively in the low micromolar or the nanomolar range in vitro, I will favor that compound to move into the animals than compound that works in the high micromolar range. And just to put this in perspective for you, the atovaquone is in the nanomolar range. Some of the compounds we've studied before are very low nanomolar range. Tafenoquine is in the low micromolar range. Quinine is in the 20 micromolar range. Clindamycin, the same thing, in the high micromolar range. Artemisinin falls in the same category as quinine. So when we do that classification, we favor compounds on the basis of high potency in vitro, but it's not necessarily 100% match between what you see in vitro in animals. That's why we sometimes go also and test drugs in mice. We have compounds like Tafenoquine and others that we are testing in my lab that work in the low micromolar range. And still, when you put them in animals, they work much better than nanomolar compounds. So there is also the pharmacokinetics, the pharmacodynamics of the compounds when you put them in animals that also play a major factor in the decision. I want also to add one more component. I know you're probably tired of my in vitro and in animal model.
Not at all.
But I want to highlight that something else that we can do in vitro that you cannot achieve right away in animals. It's doable. It's what we refer to as the therapeutic index. I know you looked at our paper and the figures, but you should look at our tables. They are really beautiful. And the tables allow us to not only compare those IC50s of the drugs against the parasite, but also the IC50 of those same drugs against human cells. And so what you're calculating is the ratio of the IC50 against mammalian cells versus that of the parasite. And that ratio is what we refer to as the therapeutic index. We define that therapeutic index as our major guide to move forward into animal studies. Again, when we look at the therapeutic index of Tafenoquine-Atovaquone compared to all other drugs, including Artemisinin.
That's fantastic. So Tom, two really important points I want to make for your audience. One, they may already be familiar with the term in vitro, but for those in the audience that are not, in vitro essentially means experiments conducted in a test tube or a petri dish. And we can learn a lot from those types of studies, but a test tube and a petri dish is not the same necessarily as a mouse, which is not necessarily the same as a human. So I think an important point that Choukri is making is this is a classic example of less is more. The less drug you can use in a patient to effectively treat that patient, the better off you are, because more isn't necessarily better. In many, many instances, using more drug is associated with a greater likelihood of adverse reactions, unintended effects or consequences, because at higher drug concentrations, you can have off-target effects that damage other cells, other parts of the body, other parts of our physiology, our metabolism, that don't necessarily demonstrate themselves in a test tube or sometimes even in a mouse. So this idea of therapeutic index and trying to figure out how little drug can you use to have the biggest therapeutic benefit, that's something that doctors should really try to hone in on when treating patients.
I think you raise a really good point. There's a lot of questions because when we take that concept and we try to bring it into our clinical practice, one of the challenges are it doesn't always work. And sometimes it appears in some cases that higher doses actually help. I've actually found that in a lot of the stuff where I thought higher doses would work, I would actually start going back to lower doses and they work better. But it's making the jump into humans, I think, is challenging. And I even know moving from a mouse to a monkey astronomically changes the cost associated with studies and the difficulty. So I know there's a challenge here, but one of the pieces where I'm always trying to how do I learn more? Because I went through, I think we talked about this previously, which is part of the reason that we're sitting here today, is that a lot of us go to medical school because we study really hard in biology and organic chemistry and all that other fun stuff that they hazed us with. And then we go to medical school and we start being told what papers mean, which then we kind of get this aura of we actually know how to read a paper. And most doctors, or physicians, I should say, MDs, DOs, even nurse practitioners and PAs, have no idea what you guys are talking about. Like even an MIC 50 or something of that, all these things we knew for 1 week in medical school, and then we assume that we actually know what it means in the real science world. But that's why we have these conversations and we have these collaborations so that, we can let you guys know what we need clinically, and you can remind us what your research really means, not what we think because a lot of us are guilty of reading the abstract and or the conclusion and then going and trying to implement this. So one of the things I think that really highlights this is, you said this before, Choukri, and I know we've all talked about this. I've seen this, and it kind of in my brain, I start to think about this whole concept of how much do I need to do in a clinical practice, duration, dosage, and stuff. But you said something that essentially, if I'm not mishearing you, that recrudescence, or kind of the relapse of someone of a mouse that has babesiosis in a non-lethal model, is essentially 100%. Is that? And I think or some treatment failure is present, right? And maybe you can clarify the actual numbers that you're seeing in your lab, because we don't talk about Babesia treatment failure. In fact, there's a bunch, until the last couple of years, politically, most people said Babesia was one and done. In fact, you might not even need to treat it. And most people infected with Babesia have little or no symptoms. It doesn't matter what their blood smear shows. It doesn't matter what their antibodies show. They're going to be fine. Unless they're not, and that's a very small number of people. And there's been this thing that I grew up in conventional medicine, that recurrence of babesiosis is rarely a thing unless you are truly immunocompromised. Yet what I think I heard you say, and what I've seen kind of clinically, is that maybe a treatment failure is more common than almost never.
So again, I'm referring to specific treatments, in this case, the Atovaquone treatment. And just to put it in perspective for you, when Atovaquone was first introduced for treatment of malaria several years ago, resistant parasites emerged within 9 days of introduction of the drug. So you have to question, how is that even possible? Well, the reason is because Atovaquone targets the mitochondria itself, and the gene that encodes the target protein, in this case, cytochrome B of the PC1 complex, is a mitochondrial protein. And the mitochondrial genome seems to be, at least in the case of Babesia microti, as we reported in our first genome of Babesia, it exists as 4 different linear chromosomes there. So through multiple combination events, you could always have accumulated multiple mutations. So in fact, we think that both in Plasmodium and Babesia, that mutation may always be there for when you treat with the Atovaquone. Now, another concept that I didn't mention today, it's very clear in our Journal of Experimental Medicine paper in 2016, is one that is quite interesting. I think clinicians and scientists would be interested to hear. There are 2 ways to do combination therapy. One is where you combine 2 drugs, and each one of them has a different target. And those targets, basically, by combining 2 drugs, you reduce the probability of resistance occurring, because you need to have 2 independent mutations occurring 2 different targets in order to resist the combination therapy. But there is another mechanism of resistance, which is based on fitness. And fitness is basically, in this case, which we think is happening in the cytochrome B, is that the mutation, why it could be there, but it's not dominant, why we don't see it all the time, it's because it has a fitness cost. So the parasites that carry that mutation are likely not growing very well. But now all of a sudden, you're killing the others that don't carry the mutation with Atovaquone, and that becomes the predominant parasite. Mm-hmm. Right? So as a result of these, drugs like Atovaquone that could become obsolete as a result of these mutations, but these mutations have a fitness cost. But they might already be there. So as a result of these, you will always see relapse as a result of drugs like Atovaquone, and this has already been noted for some time. In animals, we always see it. I mean, you can't miss a paper with the treatment of Atovaquone and you don't see relapse. We do our treatment for a window of 10 days, but we still see it. But whether we extend it to 30 days or more, we see it or not, that's a different story. The question that we are asking is this just a dormant parasite? Is this a resistant parasite that has been now amplified as a result of the treatment? Or are these dormant parasites that exist in some tissue in the host and then wakes up and starts causing infection? And we have some evidence, although the way to run these studies are quite complicated, because statistically you will need to infect many mice to be able to see this in a way that is statistically significant. We have had cases of mice that have cleared the infection for several months and then showed up with parasitemia again. And that's raises a major question. That the only reason you will see something like that in other parasites is if you have a dormant parasite. So, as an example, a very common feature of malaria amongst people who participated in the Korean War or Vietnam War, that they come home and months later, sometimes years later, they show up with malaria, with Vivax malaria. But that's because Vivax has a dormant form called hypnozoates in the liver, and then it can be triggered and it starts reinfecting. But as I mentioned earlier, they belong to the same phylum. Why some parasites choose to have dormant forms and others don't, it's a really an evolutionary event that we don't understand fully. But it's not impossible that this parasite might have a dormant form, and we just need to understand it better. Where does it occur? We don't know. Certainly, we don't have any evidence that Babesia parasite infect liver cells, but there is evidence that they might exist in the bone marrow or other tissues. So it might be there that some parasites are just dormant, and then they come back and they do it. And that's our explanation, because as I want to emphasize again, in our mouse model with Atovaquone treatment, when we see a relapse, in some cases we see a resistant parasite, we see the mutation, but in some cases we don't see the mutation at all. And those parasites, if you were to take them and put them back in a mouse and treat with Atovaquone, you eliminate the infection, and then you see a relapse again. So, but without carrying the mutation. So what do? Is it just the drug that silences them but doesn't kill them, so cytostatic rather than cytocidal effect? Or is it a dormant form and that wakes up and does it? It's really an important question, one that we want to investigate in the future. But as you know, it takes a lot of energy and effort to work on every aspect of fascinating parasites like Babesia.
Yeah. So, Tom, I'd like to follow up on one comment you made, because I think it will be helpful both to patients and to their healthcare practitioners. And this is this idea of terminology, the terminology that scientists use, like IC50s or MICs, and what the general public's understanding of that terminology is. And I want to touch on this idea of immunocompromised. Okay. When someone is immunocompromised. Now, generally, I think in the general public, when someone is immunocompromised or immunosuppressed, they're thinking of someone that may be receiving immunosuppressive drugs because they had an organ transplant, or they're immunocompromised because they're dealing with cancer or some other very serious medical condition. Obviously, we talk about being immunocompromised if you have two different infections that you're dealing with at the same time. But I think it's important for patients and health practitioners to understand that there are many other ways that an individual can be immunocompromised in a broader definition. If you're not sleeping well, you could be potentially immunocompromised, more susceptible to infection, or exhibit more severe symptoms of an infection you're currently dealing with. If you are stressed out at work, you can be immunocompromised. If you are, suffering from obesity or just in general poor diet, you can be immunocompromised. And so we said early on, what the doctors and the patients want to do is just figure out how to get someone feeling better. So there is an important, essential, inescapable role that the healthcare practitioner plays in that process of regaining health, because they're the ones that can prescribe drugs. But there is a lot that the patient can do to have agency over their health and recovering their health. And that is trying to get more sleep, eat better, exercise more. So there are a number of other factors that can contribute to more severe disease or predispose you to being challenged by other infectious agents that I think is important for patients to appreciate.
I agree. And I think I would also dovetail into that. As a clinician, I think that everything you just highlighted is like, if everyone would put in the effort and like kind of really reclaim their own autonomy over their own health and the responsibility of optimizing their own immune function through de-stressing, proper sleep, better nutrition, exercise, all that. However, I would also caution the Lyme community, if you will, because somehow we've labeled. First of all, I'd say stop labeling yourself, because that's a terrible mindset, because that's just a negative mindset for illness. But more to the point, like if you have a primary immunodeficiency, if you're on an immunosuppressant drug, if you're undergoing chemotherapy or other, that is a different type of immunocompromised than I'm not sleeping well.
Absolutely.
And the problem is, I think in the Lyme community is people read papers and they go, we're looking at true immunocompromised, like very severe models for a particular reason, but we're taking the immunocompromised word from its proper form to, like what I said earlier, there's like kind of like maybe, true bench research immunocompromised, true medical immunocompromised versus you're immuno sort of suppressed, like you're kind of, or what we might call in the functional medicine world, functionally suppressed. Like, so yes, it's interesting though, because I'm just thinking in my mind, and I didn't think we were going to go here, but I guess we will for a second. Cold exposure. It's really about one of the things that I think, and people can think about and talk to their providers about, is the concept of hormesis, where a potentially toxic threat at a lower dose for a duration that's appropriate, maybe say like cold exposure for a short period of time, maybe boosts your immune system. But if you were out in the cold too long, you would suppress your immune system and or eventually have hypothermia and die if you didn't remove the toxin. So I just think it's important to define than most people who have Lyme and Babesiosis.
Yes, I think in most instances they should be using the term immunosuppressed, not necessarily immunocompromised. And then, as a scientist, an immunologist and a bacteriologist, I have issues with thinking that you're immune suppressed because you have Lyme disease or Babesiosis, because or another infectious agent, because from a strict definition. If you're immunosuppressed, that means you're not capable of necessarily responding to the infectious agent. And what drives me crazy is, this concept that Lyme disease is a stealth pathogen. It's like, no, where is it a stealth pathogen? As soon as it gets into your body, it starts engaging your immune defenses. It's activating cells. It is causing cytokine storms. It's altering how your physiology, your metabolism, the neurochemistry in your brain is operating if you have neurological Lyme disease or neurological complications of babesiosis. So there isn't anything particularly stealthy about the relationship between the host, us, and a pathogen that invades our body or our red blood cells.
Yeah. I
Would
use
you
the term
agree with
immune
that?
dysregulation frequently to
Yeah.
kind of, because the thing is, I see a lot of people, I actually think, so many of the symptoms of these infections are actually not just like, hey, Babesia might be, altering red cells, or that might be lysis, but it's the immune reaction to these infections that's getting out of control. So I tend to, in those situations where I was just a second ago talking about immune suppression, I probably truly more commonly use the term immune dysregulation, because
Yes.
I don't really know what's going on. And what I know is the patient's having symptoms that look like their immune system is trying to fight something off and it needs help. So one of the things that, like, I think, and I think one of the reasons that, Tim, that so many people talk about Lyme as a stealth pathogen is, one, is we know there's persister forms, and so they just assume that means they're stealthy because they're hiding in some kind of other form. But the other part is, it's hard to, some of the testing is really complicated, right? And I know we've all talked about this, and I was wondering if we could talk a little bit maybe about Babesia in particular, like if, any insights on lab testing or incidents, like what are we seeing like in actual samples? Because we have this conversation about, we know that Babesia microti is by far the most predominantly known Babesia pathogen at the moment. Now, truth may change in 3 weeks when we have more studies, or 3 years, or 3 decades. But today it's like that's the one we see the most. And we are seeing Babesia otocoilii in ticks on the East Coast. Human infection has been shown a couple of times, but not really confirmed in more than 2 cases that I'm aware of. I know that we're talking about maybe in other ones, but truly confirmed in 2. And then we have this conversation about Babesia duncani, which comes up in a lot of these, Choukri, you're using these for their particular value in an experimental model. We see a lot more of it on the West Coast. We're really not seeing it in ticks on the East Coast, but people are finding it in blood tests. And so I'm just wondering if we could kind of riff a little bit and talk about what we know, what we don't know, where we might be moving forward from a testing perspective, because a lot of people are like, oh, I saw Babesia duncani pop up on my Quest or my other lab or my Igenix or whatever. And then they're like, oh, let's just hammer it with Atovaquone and Tafenoquine. But what's the reality that you guys are seeing in sort of the bench research and the epidemiological information coming across your desk?
Yeah, that's a very interesting topic. And I think one that I come to with a neutral mind, and hopefully
As we
everyone
all
can
should.
understand that there is a balance between what a clinician sees and believes and what a researcher can say, and what diagnostic centers might be providing as information. So let me just maybe give some background to your readers or listeners to clarify. My lab, in collaboration with several other labs around the world, we sequenced the first genome of Babesia microti. So all the information about Babesia microti genome, genes, proteins, everything has been released with that genome. Subsequently, many years later, we reported the genome of Babesia duncani. It's only then that you can really tell whether you're talking about the right parasite or not. Prior to that, there were specific markers for each species that you will study. That most commonly used are 18S ribosomal RNA genes, alpha-tubulin, and some other genes. That's how you will distinguish between one species and another. Now, all of a sudden, we went from 1 or 2 or 3 genes to have a collection of 4,500 genes that distinguish one species from another. So that was duncani. Right now, if you go to bioRxiv, you're going to see another paper we just submitted there, and that is Babesia divergens and Babesia Mamoun. We have sequenced their genome and assembled that genome, the best assembly you will ever find, annotated that genome. We reported every single gene, multi-gene families. We even found that Babesia Mamoun, which is a Missouri species, and the Babesia divergens with the European are actually 2 different species. Now we can reach that conclusion based on science, not about me waking up in the morning and say, it's a very nice day, I would like to have these 2 species different. Now we base it on very solid science. And that's because we have a full picture of the content of those parasites' genomes and their proteins, and we can do reconstitution of their metabolic functions, of their really how they operate and how they are likely to interact with the host. And I give you all this background to say that my dream is to continue to do the same thing for multiple Babesia species that are out there and Babesia species that we don't know, right? Mm-hmm. Now, to put this in perspective, all the diagnostic tests that were developed in the case of Babesia by different agencies, I'm not going to name any one of them in particular, have been developed on the basis of very limited knowledge. And so in this case, primarily the 18S ribosomal RNA gene, and then they use specific probes that include even major screening facilities. And if it's not micro, it's something else, and then they just tell you it's now duncani, or it's divergens, or it's otocoilii. And I can tell you from our studies in the lab, we don't trust those tests at all, just because when you put those tests, let's say by PCR, on a gel, they don't give you a clear-cut picture. In other words, you're detecting multiple bands that have nothing to do with the band of that species, but in your assay, you're getting a signal. And that signal said, oh, I got a signal. But it's the wrong signal. Mm-hmm. Right? So you're just getting noise in the background, and it tells you it's there. And what is troubling is some of the serum has been used a lot in most of these tests, where antibodies are used also. So these are just knowing whether the the individual has antibodies to a specific infection or not. And they will tell them, oh, we did a dilution of your antibodies of your serum to 128 or 1 to 65, and we see a signal. That's not good enough for us. All right. So as a result, I feel bad and I receive many, many emails, I can tell you, from patients, and I'm not a physician.
Mm-hmm.
Always to remind them that I cannot provide medical advice. But I feel bad that maybe the test that they're told is not accurate.
Yeah.
And the evidence is not strong. Now, I know that some of those organizations are realizing, and through lectures I've given over the years through Lyme Disease Association and others, GLA, I've always emphasized this point, that it's not about the fact that they are just doing this on purpose, but just because they had very limited knowledge. And now that this knowledge has increased, it's time for people to adjust their goalposts and change to better diagnostic tests. And we are working with them. Actually, we are providing them better markers, better evidence. I get emails from people upset about the fact that they have odocolia. But the reality, when you look at the samples, I've worked with an individual who sent me many, many blood smears, like over 80 blood smears, claiming that they have Babesia odocolia. It turned out not to be a Babesia odocolia at all. It's just a fungal organism there from contamination during the collection of the sample from the ear. Mm. But they would not want to listen because they're convinced that that's what the organism is. We don't do science that way.
Right.
We do science on the basis of if I had time, I could show you real images of data. When we see a signal, we're using one marker, let's say 18S ribosomal RNA gene, we immediately do another 7 other independent tests with different other genes and different markers. And even then, sometimes we are not convinced. And this is
confirm that specificity. I mean, this is incredibly important, and this translates into issues in the Lyme disease field as well. There is a lot of concern on the part of scientists, research clinicians, patients, and healthcare practitioners about diagnostic tests that generate false negative results. And that is absolutely what we should be concerned about. False negative results. So you do have Lyme disease or babesiosis or another infectious disease, and for a variety of reasons, the diagnostic test is telling you, no, you don't have it. You cannot rely exclusively on the results of a diagnostic test to make a clinical diagnosis. By the same token, I would argue that you cannot simply look at the symptoms of the patient in front of you and make a truly accurate diagnosis based on clinical symptomatology alone. You can sometimes, but not often enough. So you need that diagnostic test result to confirm your clinical suspicion. So yes, we need to improve diagnostic testing to eliminate false negatives. What is often absent or very rarely discussed in the diagnostics world, the patient treatment world, is the impact of false positive results. So you can have a diagnostic test that generates false positive results because it's not specific enough. And this is exactly what Choukri is talking about. So you don't want to find yourself at either end of that spectrum. You don't want to be told you don't have a disease when you really do, and you don't want to be told that you have a disease when you really don't. In both cases, what that does is it causes a delay in effective treatment for whatever ails you. And when you do that, invariably, maybe 100% of the time, the clinical course of disease is worse because you're allowing a pathogen, a microbe, to run rampant in your body unchecked.
Well, I think your point is so important, Tim and Choukri, both of you, because for me, tell people I really could care less what's wrong with you. Have no attachment to the name at all. However, I want to know what the name of what's wrong with you is so I can help you get there quickest. So if you want to have Lyme and Babesia, no matter what I say, just keep ordering tests, because eventually you'll find one that gives you an answer that supports your already predetermined hypothesis of what you have. But that doesn't necessarily, and then you could have chronic Lyme and Babesia for 25 years because you really don't have it and you have something else. And my goal as a clinician is to actually help you figure out what you really have and then help you get better. So if you want to get better, really pay attention to this conversation, how Choukri and his lab are working with the diagnostics companies to create the opportunity to have better testing. Because it's not about, like, you know, I feel like in the Lyme community, so much of what we talk about is just, oh, this test isn't as good, or this test isn't as good. And so we just, but then it's like, well, the test that's the best in my mind didn't get me the answer. So now I'm going to go to somewhere else. And if the B cell antibody testing didn't work, I'm going to do T cells. And if I didn't do this, I'm going to do RNA. And I'm like, we are at a place where we don't have enough accurate testing on human beings. I firmly believe we need to do a better job, as we've been talking about. And it's exciting to me as a clinician to know that the work is being done and we know more about these pathogens. We're sharing the information to it sounds like, to whoever's really truly open to find out the truth and making better testing. I guess the question, Choukri, that one of the things I don't want to forget as we're kind of winding down, and then I want to hear kind of if you guys have any closing things, but if I'm doing some of the tests that you're talking about, maybe it's I'm getting a ding on a Babesia, and you put whatever species at the end, Microti, Duncani, Atovaquone. Are you finding that these samples that are positive, say antibodies for some Babesia, are actually a Babesia in the genus Babesia, just not the species they said? Or is it just we're kind of like missing the mark completely?
Missing the mark completely.
That was not the answer I was hoping to hear.
and Global Lyme Alliance is sort of playing a small but I think important role in this process, because Global Lyme Alliance has been funding Choukri to develop better diagnostic tests for babesiosis, and we do this with other researchers around the world to develop better diagnostic tests for other tick-borne diseases. And we really focus our attention on researchers that appreciate that. You're never going to have a 100% accurate diagnostic test. But to the extent humanly and scientifically possible, you want to develop a test that has as few false negatives and as few false positives as possible.
Right.
And if I may, Tom, I gave you the answer the quickest as possible, to be very clear. I have challenged the community to send us samples over the years, as long as those samples are de-identified. And we have also, now I'm very glad that there is Lyme Biobank that actually collect those samples. There is nothing better than having a biorepository of all these samples, because we can be wrong. And I am the first one to notice this, because we don't know everything. We know a lot compared to a lot of people, but we also know, and we are humbled every day by these pathogens and what they are capable of doing. So as a result, having a biobank, a biorepository of all the samples of anyone who's been tested positive or negative or wrongly stated to be Lyme or Babesia patient, to have those samples. I can tell you from our experience, so far all the samples we have tested that were suggested to be either Duncani or Atovaquone turned out to be negative. With how many samples we test so far, we have tested close to 2,000 samples. It's not a huge collection, but we do have a plan to do a much larger study to be able to identify what it is. In the meantime, let me give you one that will maybe make your sleep tonight quite difficult and now put you in that category of immunosuppressed. We did test a lot of samples, blood samples, and we knew that they were reactive in some antigen-based detection assay. This is detecting the secretion of the antigen. And so if the antibodies recognize that antigen, we know that it is an active infection. But the titer of recognition, the signal, defines whether you're dealing with the same species or closely related species, etc. And so far, we have evidence that we are detecting some other organism that we don't know. And we are in the process, in a small number of samples, not a huge sample, but we see it in animals in about 30 samples out of about 700. And we found it in 6 samples that were already pre-screened from blood samples that were collected in the nation, without defining the organization that collect them. And all this will be reported hopefully very soon. We know for sure that it is some form of Babesia, but it's not one that we know. And we are currently sending that material. Hopefully, we don't have enough DNA in that content for sequencing, and we will have some data in the next few weeks to know how closely they are to the parasites we know, but certainly not the ones that have been claimed to be. And I would like to close this comment in particular that whether it's an organization like GLA, whether it's your organization, Tom, whether many other organizations, LDA, clinicians working, believing or not believing in the science, or us scientists doing it on a daily basis, we all have one goal, is that the patient gets better.
Yep.
And that's really something I try to be neutral, and I try to work in the middle between the clinicians who refuse to work with patients and patients who refuse to work with clinicians. I'd like to be in the middle, to use a really logical reasoning and middle ground as a way to reach the ultimate goal that everyone has, that the patient gets better, that no one gets harmed with the wrong treatment or being ignored for receiving the right treatment. We care about this a lot. When I get an email from someone and tells me, my daughter has Dankani, and what treatment would you recommend? I saw your paper, would you recommend this treatment? Or what's the evidence that it's Dankani? And they send me the data, and that's not strong evidence. They said, no, that was not my question. Would you recommend a treatment? I say, I can't. I'm not a physician, but there is some point where logic and common sense has to prevail. Because ultimately, I lose sleep when I hear from a father or a mother telling me that her child is suffering from this disease. Because frankly, even when I tell her that she doesn't have that, I wish there was a way for someone else to find exactly what that child has. Because that parent is not lying, that child is suffering. So it might not be suffering from Babesia or Lyme or malaria or something else, it might be something else. That person needs the right help. If they're given the wrong information, they're going to seek treatments that could only affect those families' financials, affect their way of living a peaceful life, and ultimately that patient is not getting the right treatment. And I just want to highlight this a lot, because we really care. If we did not care, we would not be doing this.
No, that's absolutely perfectly put. Tom, that's your drop the mic moment there. I can't say it any better.
I was thinking, like, in closing, I mean, that's exactly where I was thinking, Choukri, is like, the reason that I'm so thankful that you two spent the time to hang out with us today and to talk. My goal is not to have a conversation that everybody's happy about. My goal is to have a conversation where we talk about the truth of what we do know and what we don't. Clinical medicine is a better understanding of fundamentally what we know, the science of medicine, and then we're applying that to the patient in front of us and filling in the gaps the best we can so that we can help alleviate their suffering, which is that art of medicine. But I don't think we can throw the baby out with the bathwater just because because we don't like its hair color or, whatever, right? We really need to like understand what we know and what we don't know, because ultimately that's my goal of having this conversation, of having this podcast, sharing this information with people, is to allow them to understand that we all really care. And even when a lot of us are kind of at odds, apparently, we're really just doing because we're passionate. We want to get the right answer. And there are so many people who have, like we said, truth will change. But that is the thing there. We're not saying, but not everyone who's chronically sick has chronic Lyme or chronic Babesia. And we all really just are here to help figure out, ask the hard questions so that we move the field forward. So we do exactly what you say, which is help your child, help your partner, help yourself, find out what you really have so you can actually get better and go back to leading your passionate, peaceful, loving life, however is best for you. But if we just say, yep, everybody has Babesia based on limited knowledge, and then when we gain new knowledge, we don't adapt to that new knowledge and implement that, then people are going to be suffering longer than they need to. And so anyway, I should just not even say anything, because total mic drop. I'll definitely be highlighting that one, like, Tim said, because that's exactly why we're here. And I just want to thank you guys both for spending the time. I know to get out there and answer people's emails, to do the lectures you do, to do the hard science. I know the amount of work you both do to get to the place where you can have a conversation like we had today and putting out all those papers, and then to spend the extra time to actually kind of interpret this for sort of the physicians out there, the healthcare practitioners, the lay public, so that we have a better understanding that one of the things that I always found really interesting is the more and more I got into what was the research in Lyme and related infections, tick-borne infections, was that there are so many more people out there actually doing the work to help alleviate the suffering of our patients than most of our patients know about. And there are actually more people doing amazing research like you, Choukri, than most doctors in this field know about. It's not just us against them. This is a community where we've all come together. And if you're open to knowing who's out there, you meet two amazing gentlemen like we just talked to today. So thank you so very much for being here and for all the work you do. And you guys lead with your heart more than any two people I know. So I just want to say thank you so very much from the bottom of mine.
Thank you again for having us.
All righty. Thanks, everybody. And we'll be talking to you. Hey, and so there you have it. What an amazing conversation with Dr. Choukri Ben Mamoun and Dr. Timothy Sellati. And I really just want to say thank you so much for joining us today. This is one of my really passion projects. I love diving into the literature to find out what it really means, understand what we know, what we don't know, because I think, as Dr. Choukri said so well, is that our goal is to help each and every patient out there who's suffering with these chronic illnesses to have access to better diagnostics and have access to better treatments. And even for myself, like, I'm so excited about how truth changes, right? What I know today, even what we're talking about in this episode, may change in the near future. And when it does, I'm so excited to incorporate that into what I've been doing and changing, right? I've even, over the last couple of years, I've seen so much new research come out on herbals, basic science research means in clinical practice. I've seen lab testing get so much better. And it's so wonderful to hear about the collaboration between different labs, different departments, different companies even, so that we can all help each person who's suffering from chronic Lyme disease or other chronic illness find out, is it really chronic Lyme? Is it really chronic babesiosis? Or is it something else? Because to me, like I said, I don't really care what the true answer is in the end. I'm not attached to it. I just want you to get better. I want each physician, healthcare practitioners, whatever your degree may be, when you're helping someone suffering, I want you to have the greatest body of evidence behind you that we can, and then be able to do that freestyling, that beautiful art that allows people to truly heal when we balance that art and science of medicine. And I think a conversation today really highlighted that. And this is a great beginning, right? This isn't an end. This isn't to say somebody's wrong or somebody's right. What we're saying is we have new information that's coming out. As I mentioned in our podcast during the episode, I literally, not even 10 years ago, when I started doing deep dives into babesiosis so that I could educate other practitioners, I found out that there were literally quotes from papers that I'm paraphrasing right now, but they literally said, we are woefully underpowered. We have terrible information on Babesia. We just don't know enough. Like, they don't, and so in as little as a decade, Dr. Mamoun's lab has just really kicked up the knowledge base that we have so that then we can find out new, exciting information. And I hope that this episode really showed you that the scientific community, the clinician, health practitioner community, and the patient community all can work together. It's just so important to me that you understand that while there are personalities and different sides of this discussion, most people are coming together to that middle road, and that middle road is really focused on each and every patient who's suffering, and that's our job here. So that's why here at the Lyme Insider, we're really investigating those mysteries of chronic tick-borne illnesses, where we're really diving into how it meets that forefront of medical research. And so really, I just want to say thank you for joining us. Definitely leave us some comments. Let us know what you're thinking. Did we get it right? Is there something we may have missed that you might want to hear more about? And certainly, if you have any other suggestions for future episodes, things that you really want to know about, that's what I'm here for. In the Lyme Insider, let's dive into those mysteries. Let's find out what we know about research. Let's find out what we know from a clinical perspective. Let's blend that with the art of medicine and working with each individual so that each unique individual has the opportunity to access healing and health. And let's have the foundation be science and grow from there. So thanks so very much. This is Dr. Tom Moorcroft. Until next time, I wish you all the best.